It's 2 o'clock, let's get started. The lab exam scores aren't going to post till tomorrow because I got 28 of these to grade. And I was grading all yesterday, physiology, blue books. And I got a date tonight, so I'm not working tonight. So this will be posted, tomorrow. Tomorrow's soon enough, isn't it?
Yeah.
Okay.
Yes.
Thank God. All right, here we go. Special senses, we're going to spend most of the time on the eye and the ear. There's going to be several possible microscopic slides. Remember that for lab exam 3. Well, we're actually done using that counter for lab exams now. There will be some stations that are projected microscopic images. Those will be, I'll go over those today. But last week we just had the cells on page 25. Turn one page back. Those are the first possible microscopic slides. Pyramidal cells, cerebral cortex, Purkinje cells, cerebellar cortex. On the top of page 26, we'll see later, those are both olfactory and gustatory will be on slides, microscopic slides. On the very top of page 26.
Oh, okay.
You can see it says microscopic.
Yeah.
Everybody there? Okay. So now let's just go to the eye models, and I got all the eye models here on the cart next to the bench. This is the model that had the large eye with the lacrimal apparatus. But this muscle at the very top is the levator palpebra superioris. What does the E at the end of palpebra mean? Plural. How many can I pin with one pen?
No.
Cross off the E. Levator palpebra superioris. We already just asked orbicularis oculi, so I'm not going to ask that for lab exam 3. I asked that on lab exam 2. You could put a little star next to conjunctiva, because that'll also be microscopic. And that's basically what I do. I put a little star, some notation for things that'll be microscopic slides. So that's 2 on page 25, the 2 on the top of page 26, and now conjunctiva.
Mm-hmm.
Everybody got that? Everybody there? All right, the corners of the eye are called commissures or canthi. And one thing that'll be really good to remember is later on we'll look at the lacrimal gland. Lacrimal gland is always lateral. Lacrimal gland is always lateral.
Okay.
Lacrimal gland is always lateral, so that, you know, is the lateral palpebral commissure, or canthus. You can use commissure or canthus. But that's the lateral one because it's closer to the lacrimal gland. That means this is the medial one, in red. Medial palpebral commissure.
Okey doke.
There's the commissures there again. I rotated the model to get more of this lacrimal apparatus. So now we're in the lacrimal apparatus section. Are we ready? Best way to study this is sequential, like where do tears come from and where do they go? Tears come from the lacrimal gland. Again, that's always lateral superior corner of the eye. Lateral superior corner of the eye, lacrimal gland. There's like 10 to 12 little ductules that delivers the tear on the lateral side. It goes from lateral to medial.
Okay, so yellow is the lateral gland? The lateral?
Yellow is the lateral palpebral commissure. Same as the previous slide.
Okay.
So it's these 2 color arrows
Okay.
are these 2 color arrows.
Got it. Okay.
Okay? All right, after the tears go from lateral to medial, they go into these 2 drains. Those are the lacrimal canaliculi.
Lacrimal canaliculi. 13.
Then the tears gets collected in this lacrimal sac. And then gravity drains it down the nasal lacrimal duct. And the nasal lacrimal duct empties in the inferior portion of the nasal cavity, which is why when you cry, your nose runs. It's going in that direction. Okay?
So can you pin that if it's inside?
Lacrimal sac, nasal lacrimal duct. The drainage of tears into your nasal cavity is driven by gravity. So if you stand on your head and cry, your nose isn't gonna run. Your hair will get wet because the tears will go out on your forehead and get into your hair. But in a normal drainage into your nasal cavity, it's all gravity driven. Everybody got it?
Okay, so this
All right.
is the
Orange arrow is a separate gland called the lacrimal caruncle.
Lateral caruncle.
Uh-huh.
What?
It's right there in the middle of that section on lacrimal
Oh,
apparatus.
caruncle, okay.
Everybody got it? Now listen, pay attention. It is near the medial palpebral commissure. It is not the medial palpebral commissure. The medial palpebral commissure is in red. This is right next to it in orange.
And is it that entire circle part?
Yes, that's that pink, number 11.
Okay.
Everybody got it? Any questions? All right. This is the model for the extrinsic eye muscles. So go to the top right,
Okay.
and there's 6 extrinsic eye muscles. And you have to have 3 pairs of muscle antagonists. You guys remember what antagonist means?
Mm-hmm.
These are muscle antagonists, 3 pairs. That's the only way you can do this. You can move your eyeball in a circle. You have to have 3 pairs of antagonistic muscles. Are we clear?
Mm-hmm.
All right. Now on the very top here, there's actually 2 muscles. And you can see the crease line in between. The top one, we already saw. This is levator palpebra superioris. Mostly that you have to contract to open your eyes.
Okay.
Which probably in about 20 minutes, a lot of you are going to start relaxing.
Oh.
Okay? That's the one you got to contract to open your eyes.
Okay.
The one underneath it, that is actually an extrinsic eye muscle. That's the superior rectus.
Up and down.
Now, you might think the superior rectus muscle would pull the eyeball just straight up, but it doesn't. And the reason why it doesn't, okay, does this look familiar?
Yes.
Is because these rectus muscles parallels the optic nerve.
Mm.
And the optic nerve is diagonal. Everybody see that in the drawing? Does that look familiar?
Yes.
So, say if this is a superior view, we're looking on top of the eyeball, and the superior rectus, the yellow arrow, is paralleling the optic nerve. To pull it straight up, it has to be in this plane. But it's not, it's diagonal. And that's why the superior rectus pulls it up and in. Everybody see that? It's diagonal.
Up and in.
Down there in the bottom with the pink arrow is the inferior rectus. And it's also diagonal, and that's why the inferior rectus pulls it down and in.
Do we need to know what they do?
This is on page 26.
Well, I'm just saying, like, we need to know the name, and then do we need to add up and in? It is, okay.
Well, answer my question on the question card.
Got it, okay. Okay, sorry.
But if it's on the page, it's
Okay.
fair game.
I just didn't know if that was like a reference or a.
Okay, we'll just move on.
Okay.
All right, so the light green arrow is the lateral rectus. And the blue is the superior oblique.
Blue.
Blue is superior oblique muscle.
10. Okay.
So I still haven't covered all of them. There's only 4 extrinsic eye muscles on this slide. There's 2 more. Everybody got these?
And that green one on top, that line isn't, it isn't actually separated, right? It goes all the way to the, on the very top.
You mean this one?
Yeah.
Yeah, this is levator palpebra superioris, and it goes all the way to the eyelid.
Okay.
But on the next slide, I take all this stuff off.
Okay.
So all that stuff's off, and now you can see where the levator palpebra superioris, it's cut off right there. But now you can actually see the insertion of the superior rectus on the sclera. It actually inserts on the collagen of the sclera. That's how it moves your eyeball.
Okay.
Okay? Lateral rectus in light green, superior oblique in blue. All throughout this series of slides on extrinsic eye muscles, the color arrows don't change. All right? They're all the same color arrows.
Okay.
All right. How about we do the eye dissections next week? What do you think?
Sure.
Because this is going to take me seriously to 3:15 to finish this anyway. So we'll do this next week. But in the cow eye, the extrinsic eye muscle attachments form a complete circle. In our eye, it doesn't. Okay, everybody got it? Okay, so now I've taken off the entire structure here on top, superior rectus with the levator palpebra superioris, and all we have is the insertion of the superior rectus. That's all we can see.
Okay.
Okay, everybody got what I did here?
Yep.
Okay, now we can see more muscles. Actually, we can see all 6. So let's go over the ones we've already covered. Superior rectus. In pink, inferior rectus. In light green, lateral rectus. But here's for the first time is the medial rectus in red. We've already seen the superior oblique muscle in blue, but now here in orange is the inferior oblique muscle in orange. Now, as far as the 3 pairs of muscle antagonists, the only one that is intuitive here is the medial and lateral rectus. They're either going to pull the eyeball in or pull it out. Everybody got it? Now, because of the diagonal rectus muscles, superior and inferior, those are not antagonists. Instead, if you and just look on the top right of page 26, you got to match up the 2 superiors. The superior rectus is antagonistic to the superior oblique muscle. Superior rectus up and in, superior oblique down and out. Those are antagonists. Do you see that on page 26, or did that Rob exam just kick your ass?
No, I see that.
You can see that?
Yeah.
Same thing with the inferiors. The inferior rectus is antagonistic with the inferior oblique muscle. Inferior rectus down and in, inferior oblique up and out.
Up and out.
That's antagonistic.
Okay, I see it.
Everybody got it?
Yep.
So medial lateral antagonists, the 2 superiors antagonists, the 2 inferiors antagonists. You can move your eyeball in a circle.
Mhm.
I need to say that again. Because I thought that was pretty simple. Right? Medial lateral,
Mhm.
the 2 superiors, the 2 inferiors. There's your 3 pairs of antagonists. Now you can move your eyeball in a circle.
Okay. Are you gonna use the word antagonist?
No,
Oh,
because that's not on page 26.
okay. Just making sure I'm clear.
It's when I say something that I think is kind of simple, and I see this. And do you need me to repeat that? Because it kind of looks like I do. Everybody got extrinsic eye muscles?
Mhm.
Okay, this is where I wanted to show the entire inferior oblique. It's actually the shortest of the 6 muscles. That's the whole thing right there, from origin to insertion. But lateral rectus, inferior rectus, we've seen before. And then I can take off the lateral rectus, and now you can see a lot of that clearly. Same colored arrows. I don't need to go over that again, right?
Yeah.
Any questions? Now let's go to the eye compartments. Everybody good so far? Or at this point, can you even tell if you're okay?
I have a heartbeat.
All right, there are 3 layers or coats or tunics to the eyeball. We're talking about the eyeball wall here. 3. The 2 outer ones are complete.
Okay.
The inner one is partial. Everybody got it? The outer coat here in red is the scleroid coat or fibrous tunic. You could use either one. The one in the middle is the choroid coat or vascular tunic. You can use either one of those for the point. But read the question card. I'm either going to ask you for the structure, name of the structure, or the name of the coat.
Okay.
Got it? And then I'm going over the names of the coats now.
Yeah.
Red is scleroid coat or fibrous tunic. Green is choroid coat or vascular tunic. Purple is retina or neural tunic.
Okay, hold on.
Everybody see that right in the middle of page 26? Underline is the name of the coats or tunics. And these are the 3 arrows for the coats and tunics. Basically right here is the line. This is as far anterior as the retina goes. So to the left of this line, there are only 2 coats. Scleroid coat, choroid coat. To the right of this line is all 3 coats. Is that understandable? Everybody got that?
What's that green one? Again?
Green one is the choroid coat or vascular tunic. It's the one in the middle.
Choroid. Oh, on our, it's just confusing because on our page, they're not like in order, they're
Right.
kind of like this, so my brain
Right.
is not finding them very fast, sorry. Okay, and the 2 that are complete are the scleroid and the
Choroid.
choroid, and the retina ends
At that line.
at that bluish
Number 17.
line. 17, okay.
The retina only extends as far anteriorly as number 17. We'll see later that's called the ora serrata.
Okay.
Everybody got it? Now let's go to the structures. The scleroid coat is made up of the sclera, which is the white of the eye, which is what all those extrinsic eye muscles were inserting on,
Oh my god.
and the cornea, the clear part. It's A plus B equals C.
Sclera.
Cornea plus sclera equals scleroid coat. The weird thing is sclera and cornea is made up of the same thing,
Really?
collagen. And it's just how the collagen is put together. And if they're put together kind of scattered in every direction, it turns opaque white. When they line it up nice and parallel, it turns transparent. It's made up of the same thing. Is that wild?
And that
Okay.
plus that equals
were
that. When you say
underwhelmed.
that plus that equals that, do you mean like when they overlap, it equals that?
They don't overlap.
Oh.
This is
okay.
just in the anterior portion, plus everything in the posterior portion, sclera, equals a scleroid coat.
Okay, okay.
They don't overlap.
So, okay.
One ends and the other begins.
It's just this plus this is this, and that's how we have to do it.
Right, because the scleroid coat is the whole thing.
But that's that part and that's that part.
Right.
And together they make the whole thing.
Yes.
Okay, I'm sorry.
Yes, I'm glad I got through. All right, now, posterior to this line, ora serrata, the choroid coat is just choroid.
Okay.
So that, since now that I rotated the picture 90 degrees to get a little more detail, so here's that ora serrata number 17, and above it, it's just choroid. So that's just choroid. That's the name of the structure.
Okay.
You get anterior to the ora serrata, and it gets really complex. But this is all choroid coat. Are we ready for that complexity? Can you guys handle it? Just choroid.
Just choroid, not the coat.
Yes.
Just choroid.
And the question card will ask, what is the name of the structure?
Okay.
choroid.
Structure, not.
Same pin, different question. What is the name of the coat?
Okay.
Choroid coat.
Choroid. Right coat. Okay.
Okay. Now let's go anterior to the oris aorta and see how fancy it gets, how complex it gets. Starting from here, all the way through here is the ciliary body.
Ciliary body, 15.
There's a ciliary muscle in it. There's a gland that makes aqueous humor.
So 18 to 21, all of that.
Yeah, that's all ciliary body. The ciliary muscle has a ligament that forms a complete circle, and this ligament suspends the lens on the visual axis. So they call it suspensory ligaments, and that's in blue.
Suspensory ligaments. Okay.
On the other end of the suspensory ligament from the ciliary muscle is the equator of the lens, and it forms a ring around the equator of the lens, and the lens is the yellow arrow.
Equator.
But it suspends the lens and the visual axis. So right in the center of the cornea, right in the center of the pupil, goes right to the center of the lens. And on the back of the eyeball, there's the thing called the fovea centralis, and it's on that same line.
Oh, is that the yellow one?
Yellow is lens.
Lens, okay. Okay.
Everybody got it? This is all choroid code. I told you it got complex, didn't it?
Yeah.
Because we're still not done. Orange arrow is the iris.
Iris.
The iris has constrictors and dilators. What kind of smooth muscle is the iris?
Oh,
Multi-unit smooth muscle. Constrictors and dilators. And that constriction and dilation controls the size of the pupil. They're in purple. Purple pupil. Say that fast. Okay, you gotta read the question card, people, because I cannot suspend a pin in midair. Everybody understand that?
Yes.
I'm either gonna pin the edge of the iris and say, what is the name of the structure? It's iris. Or I can say, what is the name of the hole where this pin is placed at its edge? And the name of that is pupil.
Okay. Okay. Got it. Got it. Got it. Got it.
Okay, just a close up of the same thing. On the outside of the iris is a lot of pigmented cells. That's how it gives you your eye color. That's actually the color of your iris. Okay, green arrow at the very top is new. That is on the oris aorta.
Oris aorta.
The oris aorta is over there on the retina because it is the anterior extension of the retina. That's as far anterior as the retina goes. So behind that line, there's 3 coats. Anterior to that line, there's only 2.
Got it. Behind it.
Okay? All right, now let's go to the cells of the retina. Oh, by the way, you probably want to put a star next to cornea, ciliary body, suspensory ligaments, lens, iris, pupil.
It's a lot of stars.
All right. The cells of the retina, and by the way, this is one of the things that if you're going to take physiology that you should be studied up on from anatomy, and that's the cells of the retina. At the end of the semester, I'll tell you all the anatomy you should concentrate on for physiology. This is one of them, cells of the retina. The cells of the retina are arranged in a counterintuitive fashion. Can anybody tell me what that means?
Counterintuitive, like opposite.
It's the
than when it would
opposite of
go.
what you would expect.
Yeah.
right? So you have multicellular layers of the retina. You'd think the photoreceptors would be on the surface, pointed toward the lens where the light's coming from, wouldn't you?
Yeah.
It's the opposite. This is the direction of the light. These are the photoreceptors. They are the deepest layer, and they're pointed away from the lens. That's counterintuitive, isn't it? The light has to go through fibers of the optic nerve, ganglion cells, endocrine cells, bipolar cells, horizontal cells to finally get to the rods and cones, which are pointed away from the lens.
Is it because it's like a mirror?
It's
What's
not, well,
that?
that's how sensitive the rods and cones are. They're in the shade of all these other layers, and they're pointed away from the lens.
Huh.
That's how sensitive they are. Rods are bigger, cones are smaller. So the red arrow has to be on cones, green arrow has to be on rods. They're bigger. Okay? Now notice the shape of this at number 44 that the orange arrow is on. This is the bottom half of the fovea centralis.
Bottom half of the fovea.
So take this picture, reproduce it, and flip it so that there's now the top half. Now you have the whole fovea centralis. Do you understand what this picture is? There's only the bottom half of this. Remember fovea capitis on the femur? There was a little divot on the head, wasn't it?
Yeah.
That's what fovea means. Little divot. On your retina, there's a little divot that's in the visual axis. So if you go through the center of the cornea, the center of the pupil, the center of the lens, boom, you hit the divot on the retina in a straight line. It is the spot of highest visual acuity. So I would put a little star next to fovea centralis because it is a little divot. So everybody understand what you're seeing on this picture is just the bottom half of the divot?
Uh-huh.
Everybody got that?
Bottom half of the.
And the reason why the retina is thinner here is because at that spot, all these covering layers are pushed off to the side. That right there is the only place you've got bare cones. Because all these other layers are pushed off to the side. Can you see that? Right at this spot where the orange arrow is, you've got bare cones.
Oh.
Are the words coming out of my mouth making any sense?
No, I get that.
Okay. I'm not speaking Latin or anything? Okay. All right, so here we go. Rods and cones. Cones in red, rods in green. They synapse with bipolar neurons, also called bipolar cells. You can use either bipolar neurons or bipolar cells. Remember the two poles coming out of the cell body?
Mm-hmm.
We saw this 2 weeks ago. In fact, I used the same slide for bipolar cells.
Yeah.
Bipolar neurons are rare. We only see them in the retina and the inner ear. Bipolar neurons synapse with ganglion cells. And if you understand this, this will be a lot easier to remember. It'll be a lot easier to remember for physiology. Look at this ganglion cell right here at the bottom. Everybody see that?
Mm-hmm.
Multiple dendrites, one axon. What kind of neuron is a ganglion cell?
Multiple dendrites, one axon.
Multipolar neuron. It's a multipolar neuron. Everybody listen to me, because this is really simple. And I make it sound simple, and it's not simple. So just because I make it sound simple, don't hardly even listen because, oh, that's easy. The axon of this multipolar neuron is cranial nerve 2, the optic nerve.
What?
This is a multipolar neuron we call ganglion cell. Multiple dendrites, one axon. All these axons from ganglion cells, number 37, is the optic nerve.
Do they like fuse together, or are they
Uh-huh.
all like
Well, they don't fuse together,
They-
but they travel
they
together.
travel together?
And eventually, okay, look at this drawing. We called this the optic nerve, didn't we? That's all the axons from all the ganglion cells of that eyeball.
So that 37 is that long.
Well, it's all, so you have to gather all of them together,
To
and
get
they form
that.
this trunk.
Well, this is the bottom half of it, so isn't that all of them?
So for that whole eyeball, they're all gonna gather
And
and
do
go
that.
out here. But they're all axons of the multipolar ganglion cells.
Wow.
So, for you to see this slide, the light has to go through fibers of the optic nerve, 37, ganglion cells, 38, bipolar cells, 39, to finally get to the rods and cones that are pointed away from the lens.
Are we gonna see any examples of that, or is this our example?
Well, I'm just trying to explain this because you know why I'm explaining it like this? Because this will help you in physiology if you have heard it before.
Mhm.
To hear this for the first time in physiology will kick your ass.
Mhm.
Everybody got it? You don't have to remember it, but at least you've heard it before. And when it comes again in physiology, you're going, oh yeah, Miyahara was talking about this.
Mhm.
Everybody got it?
Mhm.
All right. Down here at the bottom left on this plastic thing on top of the cart, got retina, rods and cones. Rods are bigger and green here. Those arrows on the very top, that's the light.
Um,
And the light has to penetrate fibers of the optic nerve, which are the axons of ganglion cells. You see all the dendrites?
yeah.
One axon. All the axons, fibers of the optic nerve. Bipolar neurons to finally get to the rods and cones that are pointed away from that light.
So the one thing that I'm kind of having a hard time seeing is that the rods and cones don't look super different to me.
Yeah, just remember that the rods are bigger.
Just bigger.
See these, the bigger, fatter ones? Those are rods. The skinnier ones, those are cones.
Okay. Okay.
How ironic. Maybe this should be an eye exam. How's your vision? Can you see the bigger ones and the smaller
I mean,
ones?
I can. Yeah, I can see it. But yeah. Okay.
Okay, there's that fovea centralis. Can you see how it's like a little divot?
Uh-huh.
It is tiny though. It is really tiny. And it sits right in the middle of a yellow spot we call the macula lutea. That's the red circle.
Maclia.
It's on the same line as fovea centralis.
Oh, maclia. Okay, thank you. Why did they do that?
I probably will never ask macula lutea because you don't really have a yellow spot, a model with a yellow spot. When they, you know, an optometrist looks at your retina,
Yeah.
they can actually see the yellow spot. Have you heard the term macular degeneration?
Yes.
That's a problem with that red circle.
Oh.
You got it? Okay, blue circle is the optic disc. This is where all the axons from all the ganglion cells in that eyeball are gathering to exit the eyeball.
Mm.
You must call it optic disc. That's what the anatomical name of it is. It is functionally a blind spot because in a way it's opposite of the fovea centralis. The fovea centralis, all the fibers have been pushed off to the side. That's why it's a divot. All the fibers have been pushed off to the side right here. All cones,
Okay.
no rods, all cones, no fibers. Fibers got pushed off to the side. That's why it's a divot.
Okay.
Tracking me? Are you tracking with me?
Yeah.
The optic disc is the opposite. It's all the fibers and no photoreceptors, which is why it's a blind spot. Okay, now this model doesn't really orient it really well. This orients it better. On this slide, right retina. You're looking at the right retina.
Right retina.
You're looking at the right retina.
Right retina. Not that orange thing though. This is the right.
The
retina.
orange, yeah, the whole thing
The
is the
whole
right
thing.
retina, of which the orange arrowhead is on the fovea centralis, and it's in the middle of the macula lutea.
Okay.
Everybody see that? Now look at my right eye. If this is my right eye and you're looking at the right retina, that means the optic disc is medial. Everybody see that?
Yeah.
Optic disc is on the nose side. It's medial. Get out a piece of binder paper. Get out a piece of binder paper, split it in half, and give half to your neighbor.
Here you go, buddy.
Come on, let's do it. You guys are getting too passive. No. Horizontally like that, yeah. Just split it in half, give it to your neighbor. If there's an odd person out, I need half. I gotta do something here because you guys look bored as shit right now. Okay, here's the weird thing, and I've already mentioned this to you. Your brain sees nothing.
Yep.
It only receives action potentials, and if it goes to your occipital lobes, it generates an illusion of an image. Here's one proof of this. The refracting media, which is bending light, of your eye, cornea, aqueous humor, lens, vitreous humor. Bending that light totally inverts the image on your retina. Up is down, right is left. Right now, if you're looking at me, my head is actually on the lower part of your retina. I am upside down on your retina. Isn't that wild? Mm-hmm. You can wear glasses that re-inverts it so that up is up and right is right. And if you wear those glasses, you know what happens?
You fall over?
Yeah, you're effed up.
Yeah.
You know, you can't hardly even keep your balance for about 3 days. And your brain flips the image so that up is up and right is right again. And it takes about 3 days. And you're good as golden until you take the glasses off. And then you're effed up again for about 3 days. In about 3 days, your brain re-inverts it. Is that wild?
That's really wild.
Well, here's the thing. The blind spot is medial, so if it's inverted in your visual field, it's actually lateral. Okay? So, on one of the light blue lines, just put a small plus sign and a dot on one of the light blue lines about 1.5 inches, 2 inches apart.
Oh.
Just be sure it is on a light blue line.
A plus sign and a minus.
A plus sign and a dot.
Oh.
Make the dot small.
Okay.
But make sure it's on the light blue line about 1.5 inches, 2 inches apart. That's not even 1 inch apart. And I'm sure there's a male joke in there somewhere, but. Okay, everybody got that? Now, if the blind spot is lateral, and I'm holding up this, I'm going to close my left eye and make sure I'm using my right eye, and I have to concentrate because your superior colliculi, you remember visual reflexes, doesn't want things to stay in your blind spot. It's an unconscious reflex. Your superior colliculi doesn't want to do that. So you have to take control of these 6 muscles. The extrinsic eye muscles are skeletal muscle, and you can override the superior colliculus. You've got to concentrate and focus on the center of the plus sign. And if this dot is this far apart, about right here it disappears.
Okay, wait a minute here.
Remember, I got this close, so you got to get it closer. Because these two are pretty close together on your retina. You got to keep focused on the center of the plus sign. Your superior colliculi is going to want to make your eye move just a little bit so nothing stays in your blind spot.
Would they look like 2 dots together?
No. You're going to see the plus sign and the dot's going to disappear.
Okay. It's done.
And it's lateral. Did you make it disappear?
Yeah.
Who got it to disappear? Who didn't get it to disappear? All right, keep concentrating. And if it's like about this far apart, you're going to have to get that pretty close. I have to get it about right here to get it to disappear. About right here. Everybody got that?
Oh, yeah.
Let me ask you a question. Why don't you notice your blind spot?
Your eyes feel
It's because you have two eyes.
Oh,
It's that
goodness.
simple.
It's that simple.
The blind spot for one eye is a visual area for the other eye. When both eyes are open, you don't have a blind spot.
So if you were blind in one eye, though, it would be.
But okay, close one eye. Okay, this is gonna freak you guys out. Close one eye. Do you see a dime-shaped black hole in the lateral field of vision?
Oh, say that again?
If your brain actually could see things, you would have a dime-shaped black hole in your field of vision, just lateral to where you're focusing.
Uh-huh.
Do you have that?
Yes.
No, you don't have that.
Well, I could see some blackness.
Not
Is that what you're
unless you
talking-
took some shrooms before left.
I told you.
Why don't you have a dime-shaped hole if you actually do have a blind spot with one eye open? It is because your brain's seeing nothing. It is generating an illusion. So it's taking everything around the dime-shaped hole and just filling it in an area that it can't see. This is going to freak you out. Do the same thing again, and this is why I had you put it on a light blue line. When you do it right, you can get the dot to disappear, but notice the light blue line is totally uninterrupted.
Oh, I can't get it to disappear again.
Your brain is connecting the light blue line through your blind spot.
Oh my gosh.
Okay, let's get back to this. I needed to wake you up with some activity because you guys look bored as shit. Okay, everybody got?
Yeah.
Isn't that freaky though, how our brains actually don't see anything?
Yeah.
It's just generating an illusion. All right, we're at the very bottom of page 26. We're going to talk about the eye cavities or compartments. This is one thing that I did as a student is I needed to get this stuff in my brain unconfused in a few minutes. I averaged 4 labs a week, 15 and a half units a semester. I didn't have time to bullshit. I needed to get it in my brain unconfused in minimum amount of time. Now, is there anybody here that's interested in doing that?
Yeah.
Or would you rather just waste time?
No.
Okay. For the larger spaces of the eyeball, you have 2 words, compartment or cavity. Everybody see that?
Yes.
I always said compartment because it was a bigger word, and these are bigger spaces.
Compartment.
Especially in this section where we have confusing repetitive names. So, first of all, the main space division is ciliary body and the lens. Everybody see that? The big spaces are posterior compartment. Or if you're anterior to this line, it's the anterior compartment.
Anterior compartment. Posterior compartment. Cool.
Dividing line is a lens. But now you've got these repetitive, confusing words. In the anterior compartment, you have an anterior chamber and a posterior chamber.
Anterior.
In the anterior compartment. And now the dividing line is the iris. If you're anterior to the iris, you're in the anterior chamber. If you're posterior to the iris, but still anterior to the lens, you are in the posterior chamber. In other words, you have a posterior chamber in the anterior compartment.
Okay.
If you just do that consistently, every time, you are unconfused. You can move on and study something else. Or not, and you're going to get all confused. And what, anteriors and posteriors all over the place. What the hell? Divide it first in the big compartment. than only be in the anterior compartment to talk about chambers. Got it?
Yep.
Any questions? Does this help at all?
Yes.
Okay, on top of the card is an eyeball on a stick. Let's see the stick down here. You got the sclera and cornea. A lot of this is review. That red arrow would actually be the posterior compartment,
Posterior.
because there's in our eyeballs there's two bags of jelly called vitreous bodies and vitreous humors in there. That's really what it's on, but it's not on page 26.
Okay.
So just call it posterior compartment. The vitreous humor, by the way, gives your eyeball its shape. Oh, look what we got here, though. Anterior chamber and posterior chamber.
Okay.
And both of those are part of the anterior compartment.
And that would be orange.
This is posterior compartment.
Is that orange one the anterior compartment?
No, the orange one is cornea.
Okay. So where's the compartment in this picture? It's just showing that those chambers are part of it, but they're not
Yes.
pinning the compartment.
These are the chambers,
Okay.
which is all part of the anterior compartment.
Got it. Sorry.
Okay, you think this is stupid, right?
No, it's helping.
I'm telling you, if you just did that a few times, you are unconfused. You don't mix it up. You don't make silly mistakes on an exam, which afterwards you're all pissed off at yourself for making a silly mistake. Everybody got it? Doesn't matter what you know, not now. It only matters what you can show that you know. And if you make silly mistakes, you look the same as someone who doesn't know.
Totally.
Okay, everybody got these?
I'm sorry, can you repeat the top red one? You
That's
said
sclera.
Sclera, and the orange one's cornea?
Cornea.
Okay.
A plus B equals scleroid coat.
Okay. Sclera, cornea,
Everybody got these? Yellow is lens.
okay.
Orange is iris.
Iris.
Purple is pupil. Purple pupil. That red is ciliary body.
Ciliary body. God, I can't find these on here. Where do you find these?
All right, we're done with page 26.
Where did anyone find ciliary body on our reference?
Ciliary body is second thing under choroid coat.
Okay, thank you. Ciliary body.
Everybody done with 26? Are we good to go on? Would you like me to break now, or would you like to just finish? Who votes for finish? Oh my God. Okay. On page 27, we're going to go over the year. Get used to the number 3.
Okay.
There are 3s all over the year. For example, there are 3 main sections of the year. Look at it, underlined. External ear, middle ear, inner ear.
Okay, 1, 2, 3.
Everybody got it?
Mm-hmm.
External ear first. This is the pinna or auricle in red. Green is the external auditory or acoustic, and now you want to say canal.
Oh, okay, not meatus.
Because now it has cartilage, skin, and earwax.
Can we still say auditory?
Auditory or acoustic.
Okay.
But your third word should be canal. External ear ends at the tympanic membrane. That's the yellow arrow.
Okay.
Tympanic membrane separates middle ear from external ear. You're on one side of it, external ear. You're on the other side of it, you're in middle ear. Everybody look at me. And you gotta say tympanic membrane. eardrum gets you a half point.
Okay.
Misspell eardrum, you get no points.
It's not
Everybody
even,
got it?
It's not even on here.
Oh, I should put this up. Same color arrows for this side here. A lot of these wall charts are beaten up severely. Like, I got parts on this guy. This used to all be on there. Everything in this lab's turning to shit. Okay, everybody got these three?
Yes.
Three.
3 per sphere. Same colors as before.
All right, so here's the tympanic membrane, and it is actually, unless it's totally caked with earwax, you can actually see through it. It's so thin. You can actually see the ossicles, at least the malleus, through the tympanic membrane. And that's the malleus right there.
Malleus.
And that's the incus in blue.
Oh my gosh. Hold on.
And what's broken off on this model is the stapes. So guess how many auditory ossicles we have?
3.
3. Malleus, incus, stapes.
What was the green? The previous slide?
That was malleus.
Malleus.
And the blue is incus, right?
Incus is blue, stapes is broken off.
Okay.
Tympanic membrane.
Okay.
Everybody got this? Here's the 3 auditory ossicles. So this is, I think, on the bottom shelf. I tried to put all the ear stuff on the bottom, but I run out of room for the ear stuff. So there is a few things of ear things on the top. The rest of it is eyes. I try to keep the parts organized. Brain endocrines here, sentries here. Every week I come in here and things are moved all over the place. Like somebody here don't care.
James.
Because I mean, if teachers are disorganized,
That
how are we
is
going to help
the,
students to get organized?
That is the
Just say.
tympanic membrane.
Okay, we're still in the middle ear. There are 2 muscles in the middle ear that help protect your inner ear if you ever come see Smoking Gun Split, because we are loud. This is the tensor tympani muscle in yellow.
Tensor tympani muscle. Tensi.
The other muscle is the stapedius, and I don't have any models of stapedius.
Cool. Tensor tympani. God, that's.
See, you guys are kind of getting it. See, you guys are improving as students. Now, I just say that one time. Yeah, I don't have any models of stapedius. It's tiny, by the way, really, really small. But I can pin a tensor tympani muscle. It parallels the auditory tube. They're in green.
Okay, auditory.
You can call it eustachian tube if you want. If you have some sort of fetish about spelling Armenian names, go ahead.
Is that an Armenian name?
It's the auditory tube.
Okay.
We'll learn in the lecture what it's about. Right now, let's just get these arrows done. Okay, these 2 are the border between middle ear and inner ear. This actually is a model of the inner ear. And the green arrow is on something that's oval. What do you think that's called?
Oval window.
Oval window.
Oval window.
The red is on something that's circular. What do you think that's called?
Round.
Round window.
Round window.
So those 2 windows are the border between middle ear and inner ear.
Where does it border? Like right there at the neck of it?
What is?
Does it border at the neck of it?
Both of those circles, the oval and the circle, are the border.
Oh, they are the border.
Yeah. See, so like right here off the screen,
Yeah.
that would be middle ear. On the other side of those,
Okay.
you're in inner ear.
Okay. I thought you meant the 2, in between the 2. Okay.
Okay. This is a model of the inner ear. So we're done with any questions on middle ear. The entire inner ear is encased in a thin, bony shell we call the osseous labyrinth, and that's the orange arrow. It is actually technically part of the temporal bone.
Osseous labyrinth.
Osseous labyrinth. Osseous labyrinth for the full point.
Osseous labyrinth.
Bony capsule for the half point.
Okay.
So in the inner ear, guess how many sections of the inner ear we have?
3.
3, and they're all interconnected. Okay? Paralleling the osseous labyrinth in orange is the membranous labyrinth in blue.
Okay.
And they're, again, all interconnected.
I gotta learn how to spell.
You also have to know the names of the fluids inside these structures. Inside the membranous labyrinth is endolymph. And it's membranous labyrinth for the full point, tubular sac for the half point, which is probably a fun name to tell your friends, call your friends when they're acting weird. You do a tubular sac.
Okay. The fluid inside the membranous labyrinth is the endolymph.
Endolymph.
Okay. And
The fluid outside the membranous labyrinth, but inside the osseous labyrinth, so it's in between the labyrinths, is perilymph.
okay. Outside the membranous, in between
On the test, you would just ask the
the
question.
osseous labyrinth.
What's that?
On the test, you would just ask the question, what is the fluid in,
Yeah, I will actually pin the membranous labyrinth, and I'll either say what is the name of the structure, membranous labyrinth, or what is the name of the fluid inside the structure, endolymph.
Okay.
Or I'll say what is the name of the fluid outside the structure, perilymph.
So it's outside the structure, is it also outside the other labyrinth?
No, it's in between. So there's, see right here, this pink,
Uh-huh.
that would be perilymph.
Oh, okay.
It's outside the gray, inside the white,
Got it.
between the labyrinths. Okay, are we ready to move on? All right, so here's the 3 regions of the inner ear. You got the semicircular canals in yellow,
Okay.
and guess how many of those are on each side?
3.
3. Starting to catch this thing?
Do they all have a different name?
They do, but you don't need to worry about it.
Okay.
I would say canals.
Thank God.
Semicircular canals.
Semicircular.
Green is the vestibule.
Vestibule.
Blue is the cochlea.
Oh my God.
Those are the 3 regions of the inner ear.
Vestibule, cochlea. Cochlea, cochlea. Cochlear duct or cochlea? Is it cochlea/cochlear duct?
Just cochlea.
So
We'll
can
talk
I-
about cochlear duct later when we talk about the scala media.
Okay.
So cross off the first cochlear duct, just cochlea.
Okay. So it will repeat it. Okay.
Everybody got it? That was an answer to a physiology lab quiz. And somebody spelled it chochlea. I said, that sounds like a dessert.
Mm.
That's what I wrote in red. Sounds like a dessert. Chochlea mousse. All right, here we go. Here's the semicircular canals. There's 3 of them, and they are arranged at 90 degrees to each other in 3 dimensions, which is boom, boom, and boom. And thus, the combination of those 3 gives your brain information about any rotation, gyroscopic rotation in any plane. Your brain can sense the difference. At the base of each of the 3 semicircular canals is a swollen spot called an ampulla.
Ampulla. God, I'm not finding it.
Within the ampulla, the hair cells are part of this structure called the crista ampullaris. You might want to put a little star next to crista ampullaris and hair cells.
Hair cells as well. Okay.
Ampulla, by the way, people, is a generic term for whenever you got a tube that is swollen. So where the tube is swollen, it's an ampulla. There's other ampullae in the body. There's an ampulla of the vas deferens right at the end before it joins the ejaculatory duct. It gets more swollen, bigger. Ampulla of the vas deferens. Emergency drug kits will have emergency drugs in them. They're in these little glass tubes that are swollen. They're called ampullae of epinephrine. Everybody got it?
Yeah.
Whenever you got a tube that's swollen, it's ampulla. Okay. And here's the vestibule, but you do have to know that this is the utricle
Okay.
and this is the saccule.
Saccule
Is on the left.
left,
Utricle on the right.
Utricle. Utricle saccular. Utricle.
I'm going to teach you a really easy way of keeping that straight.
Ugh, please. Utricle.
Same color arrows here.
Saccular.
43 is the utricle. I think that's supposed to be a 44 is saccule.
And we have to say utricle macula.
No, that's what's on the inside. So that would be microscopic. Utricular macula, and I don't even think I have a picture of the saccular macula, but the utricular macula, you want to put a star next to that, as well as the hair cells. Those are microscopic images. They're inside the utricle and the saccule.
Say that again?
The maculae are inside, and they're microscopic.
Okay. Okay.
Okay, you guys want to know the
No.
easy way to keep the saccule and utricle straight? Just pretend doesn't the cochlea look like a snail?
Yes.
Just pretend the snail is a boy snail, and the sac is closer to the boy snail.
Oh my gosh. Okay.
Which is ridiculous, because if you know biology, snails have both sexes. They can go out by themselves. They don't need Tinder. Okay, there's little nerves coming out of each of the ampullae, and there's 3 of them, right? On each side, left and right, each side has 3. You got a little nerve coming out of the utricle. You got a little nerve coming out of the saccule. You got these 5 little sources of nerves that come together and form this. In red is the vestibular branch of cranial nerve 8.
Vestibular branch.
5 little things come together to form that.
And the full point we need to say of cranial nerve.
Or, and I will accept this for the full point, vestibular nerve.
Oh, okay. Done.
Because that's what it's called, vestibular nerve.
Vestibular nerve.
Okay, everybody got this? Blue is cochlea. You're going to pretend it's a boy snail. That's why that's a saccule. Okay, now the cochlea, there are 3 basic parts listed. That would be the base.
Okay.
Middle would be the coils, and it turns 2.5 times around the hub.
Where
And the very end would be the apex, also referred to as helicotrema.
apex.
Apex is the top.
Apex coils base. What's that light color?
What's that?
You're only talking, the 3 are the dark blues, right?
Yep.
Not that light blue.
No,
Okay.
these 3 dark blues. Now the bipolar neurons in the cochlea, the axon, all the axons come out right here, and that's the cochlear branch of cranial nerve 8. In light blue. What's that?
Rear branch.
And you can call it the cochlear nerve for the full point.
Oh, thank you.
Because let's look at cranial nerve 8. And that's back on page 23. Just flip back to page 23 really quickly here. When I was a student, it was called the auditory nerve. That's why on old Olympus towering tops of fat-ass German, that's why I asked words. Well, now it doesn't work, because now it's vestibulocochlear. Because guess what? Only the light blue has to do with auditory hearing. Red has nothing to do with hearing. And so they decided to change it from auditory to vestibulocochlear nerve. Vestibular nerve, cochlear nerve, vestibulocochlear, this whole thing is cranial nerve 8. Does everybody see why it's called vestibulocochlear on page 23?
Mm-hmm.
Red and light blue, vestibulocochlear. All right, let's go back to bottom page 27. This model actually shows better the 5 different trunks that come together to form the vestibular nerve. This is ampulla again, where it's swollen. And the rest of the arrows are the same colors.
So 1, 2, 3. That's apex, scleral base,
Uh-huh.
cochlear nerve,
Everybody got it?
vestibule.
Do I need to go over any of these?
Can you just go through
Ampulla, utricle, saccule, vestibular nerve, base, coil, apex to the cochlea, cochlear nerve.
Okay, apex, scleral base.
Is that good?
Yeah.
All right, now we can open up the cochlear,
Oh my god.
and when we open up the cochlea, it looks like we got how many chambers?
1, 2, 3.
3. You guys catching the, there's 3 all over, right? I will always put scala vestibuli on top.
What do you mean?
I will never put the cochlea chambers upside down on you.
Oh, okay.
On lab exam 3, it'll always be on top.
Okay.
I always put scala tympani on bottom.
Scala tympani.
I always put the scala media in the middle. Well, it's going to be in the middle regardless what I do. You can call scala vestibuli the vestibular duct for the full point, I'll accept that. You can call scala media the cochlear duct. And you can call scala tympani the tympanic duct. Everybody see that? Those are fair game synonyms. You can use either one for the full point. Everybody see that on page 27?
Okay. Would you mind pointing?
Now there is a very thin, simple squamous membrane separating the scala vestibuli from the scala media, and that's the vestibular membrane. That's the yellow arrow.
Vestibular membrane.
By the way, the cochlear duct, scala media, is the membranous labyrinth. That's the membranous labyrinth. Orange arrows. These are the bipolar cell bodies. And because these chambers are going around the hub 2.5 times, there's this row of gray matter going up 2.5 times. So they call that spiraling ganglion the spiral ganglion. And that's the orange arrows. But it is the cell bodies of the bipolar neurons.
Could you start at the green and just do it boom, 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 boom real quick? Thank you. One time.
That's a shot now.
Okay. Get you drunk soon.
This is scala vestibuli, vestibular membrane, scala media, scala tympani. Isn't red.
Okay, cool. Thank you.
Anything else? Okay, so this should have been light blue. I'm gonna have to change it to light blue. The cell bodies of the bipolar neurons in here, but then the axon that came out, that's actually the cochlear nerve. So this purple arrow is cochlear nerve.
Cochlear nerve.
Same color arrows here. One of the good things about this model is it does show the scala media as being the membranous labyrinth. It has endolymph in it. Okay. This is on the bottom of the card. Same color arrows.
Oh, okay.
All those arrows we already covered.
So you
Same
can
colors.
ask it in any of these views, like hollowed out, filled in.
Huh?
Well, like, just, okay.
So look at these arrows.
Yeah, they're all the same.
They're all the
Like,
same.
this is like filled in with colors, this is colors removed, and this is hollowed out.
Yeah, okay.
But
I
they're
see
all-
what you're saying, yes.
Okay.
It is hollowed.
So you can ask it in any of those.
Any of those. And you can see here, though, that these are the cell bodies, and this is the dendrite of the bipolar neuron, and this is the axon of the bipolar neuron, and the axon of the bipolar neuron is the cochlear nerve.
Okay.
Okay, everybody got this? This model is really good because it shows a simple squamous vestibular membrane. It is so thin, sound waves go through these two chambers at the same time. Because sound waves go right through that simple squamous. Okay?
Okay.
Same color arrows here. Everybody got those?
Mhm.
We're almost done. In the box is the spiral organ. It used to be called organ of Corti.
Oh, in the box itself.
This is the spiral organ.
Okay.
It sits on a thick basilar membrane, here in purple.
Basilar.
Sound waves don't go through this thick membrane like it goes through this thin vestibular membrane. See how they made that simple squamous? Isn't that nice of them?
Basilar.
The hair cells are in light blue. And they're called hair cells, but they don't have hair, all right? They just look like they have hair. They're actually cilia. And in all three regions of the inner ear, the cilia are embedded in something that's like jelly. In the cochlea, the cilia are embedded in this jelly-like membrane called the tectorial membrane. It is the only membrane here that is not complete. This completely seals off two chambers. This completely seals off two chambers. This one does not.
Tectorial membrane.
Tectorial membrane is like a jelly patio roof. Doesn't that look like a patio roof?
Yes.
Okay, any questions before we go to the microscopic slides? Here's our chance to confirm what you've starred. Are we ready? Turn to page 26, go to the top. And there's your olfactory epithelium. Now, a lot of textbooks will show olfactory epithelium with the free space on top. And that's nonsense, because olfactory epithelium is not on the floor of the nasal cavity, it's on the roof. It should be oriented like this. Everybody see that?
Okay.
This is going to go back to our study of the skull. Olfactory epithelium is on part of the superior nasal septum. This is nasal septum. The roof of the nasal cavity, which is tiny, and a little bit on the superior nasal concha. Look how small that is. That's why I couldn't pin superior nasal concha. It's tiny. We have very little of olfactory epithelium, which is why our sense of smell is stunted compared to other mammals. Which, if you live with other human beings, it's probably a good thing. Okay, everybody got this?
Mm-hmm.
This is how all slides of olfactory epithelium should be oriented, with the free space at the bottom, because this is the roof of the nasal cavity, not the floor. You cannot smell with cells on the floor of your nasal cavity. Taste buds. Those are those round things.
Whoa.
And they are located around the papillae of your tongue. You do have some on your palate and some in your pharynx, your throat, but most of them are on your tongue.
What's that in the middle?
What's that?
What's that white part in the middle?
That is an open space between two papillae. So if you imagine this papilla is going like this, you guys remember what papilla
Yeah.
is? It's a finger-like projection.
Yeah.
So like there's this one finger here, and there's this other finger here.
Got it.
And that's just the crevice between the two fingers, the two papillae. Everybody got it?
Will we always see a free space in the slide for a taste bud?
Yes.
Okay.
That was actually a very intuitive question. And if you ask good questions, I'll tell you. Yes, you'll always see that crevice. All right, I had you put a star next to cornea on page 26. I had you put a star next to ciliary body. So this is still cornea. This is ciliary body. This is lens. And this is iris. How'd you put stars next to all those tonight?
Yeah.
Oh wait, I don't have an arrow, but this is conjunctiva.
So we don't have these slides because
Oh,
it,
we have that. Oh,
they,
yeah.
well, what happens is they go and then it goes on top of it.
Well, it's not on the PowerPoint handout, but if you have it downloaded on your computer,
Then we can find
you
it
have
there.
all the pictures.
Okay.
But on a handout, you can only have a picture that's on top. You guys want me to go over this?
Yes.
What's that? Yeah, are we ready?
Yeah.
Let me do it in order of numbers. Number 1 is cornea.
Number 1 is cornea. Where the hell is number 1? Oh my god.
Number 2 is conjunctiva.
Conjunctiva. Okay.
Number 3 is sclera. I probably won't ask it. I'm not microscopic.
Okay.
Number 4 is ciliary body. Number 5 is anterior chamber.
Anterior chamber. Oh.
Number 6 is iris.
Iris. Where's 6? Where's 6?
Number 7 is posterior chamber. Number 8 is posterior compartment. Number 9 is lens.
Is that the whole swoop of it?
Yeah, so that whole, it's actually going to be an oblong ball. Number 10 is pupil.
What does 10 include?
What's that?
What does 10 include? Like it's just the white
Well,
space?
okay, that number 6 is the iris. There's going to be an iris on the other side, and it's the gap between the two irises. That's the hole in the middle of the iris.
Got it.
Don't worry about 11. Well, don't worry about the rest of it.
Okay.
This is called the canal of Schlemm, where aqueous humor drains out to your tears, and if it gets plugged, then you get glaucoma, and then you can smoke weed. I'm getting punchy. This is a good picture of the cells of the retina. I probably won't use it for exam 3, but I thought it was a good reference, so I put it in. You did put a star next to fovea centralis, right?
Mm-hmm.
Look how it's a divot.
Oh yeah.
This is the whole thing. So on that one picture was just the bottom half. This is the whole thing. There's another one, the whole thing. Right where that blue arrowhead is, that's bare cones. All the other superficial layers are shoved off to the side, 360 degrees around the midline, or the middle point. You should put a star next to optic disc. These are all the axons of the ganglion cells for that eyeball.
Is it that whole black area?
It's between the green arrowheads.
It's the inner part. Okay.
The optic disc is between the green arrowheads. All right, go to page 27, and near the bottom, you want to put a star next to utricular macula. It's not only here, it's also here, and this is showing you how the three regions of the inner ear are all interconnected. They're not separated, they're interconnected. So here's a utricle, and boom, here's an ampulla of the semicircular canal. This is the crista ampularis. So put a star next to crista ampularis as well as ampulla. That's blue arrow is the crista ampularis, and the hair cells are right up here. These are the hair cells. The cilia is embedded in this jelly sail called the cupula. All right.
Oh my God.
This is basically like the last slide of a model before the microscopic slides. It's actually oriented the same way too.
Wow. Say that again?
This is exactly oriented the same as this.
Okay.
Everybody look at this model right here. You see it?
Yep. Okay.
Everybody see how it's the same?
Yes.
So scala vestibuli, scala media, scala tympani.
And that number four.
Look how thin the
number-
vestibular membrane is. That is simple squamous. 8 is basilar membrane, and 5 is spiral organ. 6 is the spiral ganglion. I think these are the same color arrows. Do you need for me to go over any of these? But pretty much everything in the under cochlea, except base, coil, and apex, you should put stars next to. Everything else could be asked in a microscopic slide. I think all except for the cochlear nerve is on here. Everything else besides base, coil, and apex and cochlear nerve is on this slide right here. On the previous slide, yeah, we don't really show the cochlear nerve, so we might want to not star the cochlear nerve either. And then, so, I know you guys have had enough, but keep this PowerPoint handout for next week. And okay, the next week is going to be kind of longish because we're putting off dissections. So this is what we're going to do next week. We're going to start off right away with eye dissections. And we're going to get this thing done hopefully within a half hour, okay? And then we're done with lab exam 3 stuff. Then we're going to start lab exam 4 material on the heart, first part of the circulatory system too. As soon as we're done with that, then we're also going to do heart dissections because we're doing the heart. And so next week we'll do 2 dissections, one at the beginning with the eyes, one at the end with the hearts, and then we're done with dissections for the semester. Any questions? Okay, that's all I got for you.